another thing which we can expect to happen is if you are dealing with an ester or an amide, that they can undergo hydrolysis by esterases or peptidases to produce the corresponding carboxylic acid. So, you have R C double bond O, let us say we have a methyl ester that is going to cleave and form carboxylic acid and methanol. Okay. You can also have R C double bond O NHR, which can be cleaved by peptidases to form R C double bond O OH, the carboxylic acid, and amine. Okay. So, of course, these enzymes are present in various organs and in nearly all cells, and so therefore, this is may not be restricted only to the liver. Okay. Now, we all know that amides tend to be hydrolyzed much slower compared to esters, and typically the presence of an electron withdrawing group. So, for example, if we have an ester of benzoic acid versus an ester of para-nitro-benzoic acid, the one with the electron withdrawing group is more susceptible to hydrolysis. This can be explained because you have usually a serine residue which is going to attack here. It is a nucleophilic addition reaction, and so nucleophilic addition reaction, the carbonyl which has an electron withdrawing group on it is more susceptible to attack because it has a greater concentration of partial positive charge that is given to it by the electron withdrawing group. So, one can imagine that this reaction is going to be faster. On the other hand, you can also have an ester which has, let us say, a CF3 group on it versus an ester which has CH3. So, between these two, the product of this reaction is C double bond O OH plus CF3 CH2 OH and CH3 CH2 OH. So, the stability of or the leaving group ability of this trifluoroethanol is much better than the leaving group ability of ethanol, and so these are more susceptible to hydrolysis. Now, let us talk about some phase 2 transformations. Phase 2 transformations are basically conjugation reactions, which means that you have two molecules which are going to react with each other and form a bioconjugate, and these are catalyzed by enzymes known as transferases. Okay. The resulting conjugates are usually inactive, but of course, there are exceptions to this rule. Okay. So, the first class of conjugation reaction is addition of glucuronic acid or conjugation with glucuronic acid. So, glucuronic acid is basically a sugar whose structure I am going to draw out now. So, it has an OH here, it has a COOH, OH, OH. So, it is basically a sugar with a carboxylic acid, there is the glycosidic bond, and you have usually you have UDP as the leaving group, okay, that is uridine diphosphate, and once this reacts with an alcohol, let us say, what happens is that you kick out UDP, which is the phosphate, you kick out UDP and you form RO with the glucuronic acid sugar. So, let us draw out the structure of the sugar and we will find something very interesting about this. So, the sugar is basically in the chair conformation. You have O R here, O COOH is in the axial equatorial position, OH is in the equatorial position, equatorial, equatorial, and so on, right. But the important point here is that the UDP, which was in the axial position, has been kicked out and the new alcohol is in the equatorial position. And so, at the anomeric center, there is an inversion that occurs. Okay. So, this is catalyzed by the enzyme known as glucuronyl transferase. Okay. So, now one important point about this enzyme or this process is that you are actually producing a highly polar glucuronic acid attached to a somewhat less polar alcohol. Okay. So, again, as you can see, the common theme here is that you conjugate it with something that is highly polar, so that you can facilitate excretion. And so, this reaction can occur with phenol, alcohol, hydroxylamines and carboxylic acids. So, we will look at some examples now, and the resulting conjugate is excreted in the urine, but also it can also be excreted in the bile if the molecular weight is over 300. So, with this, let us look at some of the examples of glucuronidation. So, a variety of functional groups can be involved in this. So, you have R C double bond O NH2, which is basically an amide, which can form glucuronide, which I am just going to call it as GLUC, right. Similarly, you can have a hydroxylamine or hydroxylamino derivative forming R N O GLUC. So, basically this is the OH that is going to substitute the and produce the sugar, and they can also be examples of sulfonates, sulfonamides reacting. So, R S double bond O NH2 can give you R S double bond O NH glucuronide. So, these are again some examples of how nitrogen or oxygen based glucuronides can be formed. You can also have an amine reacting with to give you RNH GLUC. Other examples are thiols to give you RS GLUC and so on, right. Another common form of conjugation is called the sulfate conjugation. So, here what happens is that a sulfate group is added, and this is of course far less common compared to glucuronation, and it is mainly restricted to phenols, alcohols, amines and N-hydroxy compounds. So, the transformation that we are talking about here is ROH with a sulfotransferase, okay, and the product that is given is RO S double bond O OH, okay, and this is actually catalyzed by a species known as 3' phosphoadenosine 5' phosphosulfate. Okay. So, if you look at this structure, you see here that this is the phosphate and here is the sulfate. So, it looks exactly like an adenosine phosphate, except that it has a sulfate group that can be transferred. So, once this this group is ready, the enzyme sulfotransferase uses this as a cofactor and transfers a sulfate group. Okay. Aromatic hydroxylamines and hydroxamides also form unstable sulfate conjugates that can be toxic. Okay. Now, drugs bearing a carboxylic acid group can become conjugated to amino acids by forming a peptide link. Okay. In most animals, glycine conjugates are generally formed, but glutamine is the most common amino acid used for conjugation in primates. The carboxylic acid present in the drug is first activated to form a coenzyme A, thioester, which is then linked to the amino acid. So, what happens is you have R C double bond O OH, which then reacts with ATP and acetyl CoA to give you R C double bond O AMP, which then reacts with CoA SH to give you the corresponding S derivative. Okay. And this derivative is now active and it reacts with an amino acid to give you R C double bond O N amino acid. Okay. So, this is how an amino acid can be linked to a carboxylic acid. Okay. It is transferred by acetyl CoA. And there are other reactions which can, conjugation reactions which can occur, which is basically the enzyme glutathione transferase, which transfers the tripeptide glutathione to produce a glutathioneolated adduct. Okay. There are a number of electrophilic functional groups such as epoxides, alkyl halides, which are produced during epoxides are produced during cytochrome P450 mediated oxidation. You can have alkyl halides in many drugs. Sulfonates are again possible, and of course disulfides, and there are other radical species that can be produced. Okay. And these are all targets for this tripeptide known as glutathione. Okay. So, the structure of glutathione is as follows. So, CH2 COOH N C double bond O N, and this is attached to the thiol residue, which is from cysteine. This is glycine, cysteine, right, and the last amino acid here is glutamine. Okay. So, yeah, so this is glutamine, right. So, this is a tripeptide that is derived from cysteine and is very commonly used in the cell for protection from oxidative stress as well as in these conjugation reactions. So, this glutathione is typically abbreviated as GSH. Okay. So, glutathione conjugation can happen in nearly all cells, and especially these occur quite more frequently in the liver and kidney, and it is catalyzed by the enzyme known as glutathione transferase. This conjugation reaction is important in detoxifying potentially dangerous environmental toxins or electrophilic alkylating agents which are formed during phase 1 reactions. So, for example, during the phase 1 reaction, when you are dealing with an aromatic system, you end up forming an epoxide, right, and this epoxide can then be conjugated with glutathione to produce a glutathioneolated conjugate such as shown here. Okay. And similarly, you can have Michael acceptors which are formed, which can then react with glutathione to produce these types of molecules. Okay. You can also have regular epoxides which can react with glutathione to produce the corresponding glutathioneolated adducts, or you can also have sulfates which are excellent alkylating agents which can react with glutathione to produce this kind of an intermediate. Okay. Now, not all phase 2 reactions result in increased polarity. There are some examples of methylation and acetylation which are important phase 2 reactions which can occur, but these usually result in decrease of polarity. Okay. Now, let us look at methylation reactions. Methylation reactions are catalyzed or mediated by the species called as S-adenosyl methionine. So, the example of a reaction that we are looking at is R2NH. So, there is an enzyme called as methyl transferase in the presence of S-adenosyl methionine. So, you see here that this is the active species of S-adenosyl methionine, and it gives you R2NH Me. Okay. And similarly, you can also have a thiol reacting under similar conditions to give you SME, or you can have an alcohol reacting to give you ROMe. So, all these are methylation reactions that are mediated by SAM. Okay. So, in order to look, let us spend a couple of minutes and look at how SAM functions. So, you have the S-adenosyl methionine, whose structure I am not going to draw completely, but it has an important SME group here. And now, if you can imagine that there is a nucleophile such as an alcohol ROH, it can attack over here, and this is being an excellent leaving group because it is a neutral thioether. You can propose, or one can imagine that this reaction can occur very easily, and it will give you this thioether plus ROMe. Okay. So, it is a very important reaction. Methylation is a very important reaction, but keep in mind that once the functional group or the molecule is methylated, the polarity of the molecule actually decreases. Okay. Now, one important exception is there in this case. So, we have this molecule called as pyridine, whose structure is shown here, basically an aromatic ring with a nitrogen on it. So, once this gets methylated, it forms a N-methyl pyridine, which is actually has a full positive charge. So, here upon methylation, this molecule becomes more polar, right. The other reaction that we looked at is acetylation. So, primary amines such as RNH2 can react with or can undergo acetylation reactions and form RNH C double bond O CH3, and these are mediated by N-acyl transferases. So, when you have a glutathione being transferred, you have glutathione transferase. When you have a methyl group being transferred, you have a methyl transferase, and when you have an acyl group being transferred here to an amine, it is called N-acyl transferase. And again, this is mediated by acetyl CoA, which we have already looked at in the previous slides. Okay. Lastly, it is also possible for drugs bearing carboxylic acids to be conjugated with cholesterol. So, here is the structure of cholesterol, and it has a free alcohol present here. And so, you can imagine that an carboxylic acid R C double bond O OH can react with the cholesterol oxygen to give you R C double bond O cholesterol. Okay, so, this is a drug with an ester can do a transesterification reaction, wherein cholesterol can be added. Okay. Sometimes an alcohol functional group can also form conjugates with other fatty acids to produce an ester functional group. But again, all of these transformations are going to result in increased hydrophobicity or reduced polarity. Lastly, we want to discuss about metabolic stability, where ideally a drug that we are going to develop should be resistant to drug metabolism, because the production of metabolites usually complicates drug therapy. So, you have a lot of side effects associated with this. So, an example with wherein this metabolite is going to be problematic is in paracetamol, which is very commonly consumed. And what happens in this paracetamol is that it forms a metabolite which is actually quite toxic, and it can cause it also cause some of this can have carcinogenic properties. It is also important to recognize that among humans there are differences in the race of a human, and this can also manifest itself in how a drug is metabolized. So, sometimes certain drugs act in a particular way or metabolized in a particular way in white or Caucasian people, and it act in a very different way in Asian or in Chinese people, and therefore, this is also important factor in determining the metabolic stability.